Hello everyone, welcome to the Biology Funda. I am Prapti Nayak, your biology educator and founder of the Biology Funda. This is a continuation of the last video where I was teaching cell, the unit of life. If you haven't watched that video, please do watch. Next, we are going to learn about the prokaryotic cell. Prokaryotic cells are those cells which are unicellular. They do not have a well-defined nucleus and they lack membrane-bound organelles. They consist of bacteria, blue-green algae, mycoplasma, and PPLO, which is pneumonia-like organism, which are species of mycoplasmas. If we look at the shapes of the bacteria, they are divided into 4 types based on the shapes. If the bacteria is rod shaped like this, it is called as bacillus. If it is spherical, it is called as coccus. If it is like a comma shape, it is called as vibrio. If it is spiral, it is called as spirillum. Now, prokaryotic cells are smaller than the eukaryotic cell and they multiply more rapidly than eukaryotic cells. If you look at that cell wall, all the prokaryotic cells have cell wall except in mycoplasma. They also have cell membrane and there are some extension of cell membrane called as mesosomes. The cytoplasm is present, which is a semi-fluid matrix of the cell. They do not have a well-defined nucleus. The genetic material is naked and it is in a circular DNA form, single chromosome, and can be called as nucleid, not covered by a nuclear membrane and obviously called as nucleoid. Organelles are absent except ribosomes. Beside this DNA, which is naked, there is another kind of DNA, small circular DNA called as plasmid. This plasmid contains certain unique phenotypic characters and these play a very important role in the bacterial transformation as they are antibiotic resistant. The gene is used for the bacterial transformation and this you are going to learn in the chapter biotechnology in class 12. Prokaryotic cells have a unique feature called as inclusions, which we will learn in this video in detail. Now this is a prokaryotic cell. If we look at the outer structure is called as cell envelope. And this outer structure consists of 3 layers. The outermost layer is known as glycocalyx. The middle layer is called as cell wall. And the innermost layer is called as the plasma membrane. Now this glycocalyx, if it is loose sheath, it is called as slime layer. Loose sheath, that means it will retain moisture. It helps the bacteria. It prevents the bacteria from drying. Next, if it is tough and thick, the glycocalyx is called as capsule. If you have seen the capsule medicine, you see it is very thick and the content which is there inside it cannot be leaked because it is very thick. Likewise, this capsule help in the protection of the content inside the prokaryotic cell. Next, middle layer is the cell wall, which is rigid and it is absent in mycoplasma. What is the function of cell wall? It gives shape to the cell and it also protect the cell from collapsing, bursting. Then lastly we have plasma membrane, which is selectively permeable. Since it is flexible, there are some extension of plasma membrane which you will find in the cell, like mesosome. Mesosomes are extensional structure. There are three structures: tubule, vesicle and lamella, which help in cell wall formation, DNA replication and distribution to daughter cells, respiration, secretion processes, increase in the surface area of plasma membrane, and it increases the enzymatic content of the cell. So this help in the more ATP production. Now in cyanobacteria you will find another kind of extension of plasma membrane called as chromatophore, which contain pigments. These pigments help in photosynthesis in cyanobacteria. Then you will find other structure like pili, fimbriae. They have no role in the motility. They will not help the bacteria to move. Then what is the function of it? This pili, it helps in the conjugation, that means transfer of DNA. Like if there are two bacteria, through pili the DNA is transferred to the other bacteria. This is another kind of sexual reproduction in bacteria. It's a type of sexual reproduction in bacteria, not totally a sexual reproduction. Then we have fimbriae that you will find in prokaryotic cells, which are small bristle like fibers which attach bacteria in streams and to host tissue, while these pili are the elongated tubular structure made up of pilin proteins. Flagella are made up of flagellar proteins. Like this is a flagella, which is a extension of the plasma membrane, which is an outgrowth of the plasma membrane, and it is made up of three parts: basal body, hook, this is hook, and the last part is the filament. Beside that there are plasmids, ribosome, which is 70S, that we will learn further. Then there are some inclusions which are unique characteristic of prokaryotic cells. Now based on staining, bacteria are divided into two types. If this bacteria is taking the stain, it is called as gram positive bacteria. If it is not taking the stain, it is called as gram negative bacteria. This was discovered by Christian Gram. Let's learn about the ribosomes. Ribosomes are those organelles which does not have a membrane. They are found in prokaryotic as well as eukaryotic. In prokaryotic, the ribosome found is 70S. In eukaryotic, the ribosome found is 80S. These ribosomes are made up of two subunits, the larger subunit and the smaller subunit. In prokaryotic, this larger subunit is 50S, smaller subunit is 30S. Total is 70S. Do not add them. It is not based on the addition. In eukaryotic cell, the larger subunit is 60S and the smaller subunit is 40S. 70 plus 40 is not 100, but here the overall is 80. So what is that S and why it is not based on the addition? S is Swedberg's unit, which is a sedimentation coefficient, and it indirectly measure a density and size. Suppose you take the ribosome and put it in the test tube and then you go for a centrifugation. The rate at which it will sediment is the sediment coefficient. So the larger subunit of prokaryotic cells sediment at 50S. The smaller will sediment at 30s. If we put them together, it will sediment at 70s. So it is not the addition. These ribosomes are composed of RNA and protein. If you want to learn the detail in larger subunit of prokaryotic cell, you will find 5 sRNA, 23 sRNA and 31 proteins. Where in smaller subunit, you will find 16 sRNA and 21 proteins. If you look at the diameter, the length, it is 15 m by 15 nanometer by 20 nanometer. Well, if you look at the RNA and proteins in eukaryotic cell, in 60s it is 5 sRNA, 5.8 sRNA and 28 sRNA, and there are 49 proteins. While in smaller subunit you will find 80 sRNA and 33 proteins. So these are granular structure and they are discovered by George Pellet in 1953. Since they are discovered by George Pellet, they are also known as Pellet particles. If we look at the ribosomes, there are some ribosomes which will attach to a single mRNA. This structure is called as polyribosomes or polysomes. Next, we will learn about inclusion body. What are inclusion body? They are the reserve material in the prokaryotic cells. Where they are available, they are freely available in cytoplasm. They are non-membranous. That means they will not have any membrane. Examples are phosphate granules, which will store phosphate, synophysin granules, which will store some pigments, glycogen granules, that means they will store glycogen. Then there are gas vacuoles. Gas vacuoles are like gas bubbles, which you will find in blue, green and purple green photosynthetic bacteria. What is the role of it? They will not allow the bacteria to sink, so they maintain the buoyancy of the prokaryotic cells. In the next video, we will learn the further parts of this chapter. Till then, subscribe our channel, like this video, share with your friends. Thank you so much. Dev Bare Guru.